But let's start with introductions. So if we can please start, Dr. Weimer, can I ask you to start off the introductions, and then we'll come on down and say who you are and your clinic affiliation?
I'm Dr. James Weimer, sitting in Dr. Tohidi's space, and I will move over when he shows, but I'm at the University of Florida in Gainesville. Okay, as everybody knows, I'm a Gator. Okay. I should say I'm also at the VA. I should also comment. I spend as much time with veterans at the VA.
Hi, I'm Dr. Michael Pulley. I'm at University of Florida in Jacksonville, and I'm the director of the center there.
Good day. I'm Bjorn Askerson. I'm at Mayo Clinic, Florida in Jacksonville.
Dr. Doro Locatelli from the Nova Southeastern University, Kathy J. Hasman ALS Center.
Hello, Amy Chen, director at the USF ALS Center.
Hey there, my name's Nathan Carberry. I'm at the University of Miami.
And now I'd like to invite Dr. Amy Chen to the stage for her presentation.
Hello, good afternoon, everybody. Thank you, Elsa, for organizing this annual event in Florida. Thank you, everybody, for coming and staying. Before I start this talk, I would like to acknowledge that there are people in our communities who have experienced the misfortune due to Hurricane Helene, and our hearts go out to them. And we all know that we can rebuild a community, so we hope and help we can make that happen. Now, sorry, I didn't ask, do I advance? Okay. It is my honor to be here among colleagues and friends, and it's also my pleasure to provide you a quick overview and tutorial on clinical trials. My hope is that by the end of this 1-hour session, you have a better understanding about what clinical trials are and how you can be involved. So clinical trials, they are basically studies in humans designed to answer a specific question related to a disease. And that sounds like science or research, and they are. So why should you care, and why do you want to know about them? I'll give you 3 very good reasons. One is we need to study people who have a disease to better understand what is happening to the cells, to their tissues, and their bodies. And in this case of ALS, what is the mechanism of motor neuron degeneration? The second reason is we need to test if there are going to be better and more effective treatments to help people with the disease. The third reason is we cannot do this in a vacuum, so we need patient participation to achieve those 2 goals. And I'll give you 2 more specific reasons for why clinical trials are especially important in ALS. There are different types of motor neuron diseases due to different causes. If we want to study ALS and find treatments for people with ALS, we have to first make sure that the diagnosis is accurate. And we are sure that we're studying the disease, but not other conditions that may mimic it. Now, getting patients to an ALS Association affiliated clinic, a recognized center, or a certified treatment center of excellence is the very first and important step to make sure that patients have a proper diagnosis and they are getting evidence-based standard of care that is coming to a multidisciplinary clinic. The second specific reason for the importance of clinical trials in ALS is because we do not know why does a disease start differently in different people. And we don't quite fully understand why some people have a faster progression, some people have a slower progression. Why do we see that sometimes the disease affects people in the way that they have very stiff extremities, and in some cases they have very floppy and weak extremities? So we currently treat all ALS patients for the most part the same way, except for SOD1 gene-related ALS cases. So if we can identify what is different inside the bodies for different subgroups of patients, then we will be closer to providing individualized and targeted treatment. And that's the future of medicine, precision medicine. I hope by now maybe I have convinced some of you that doing clinical research in ALS is very important. So what are the types of clinical studies that are available to people? First, observational studies. They are basically research that is done to collect data related to people's health, their lifestyles, or exposures that they may have. And this type of study is usually very inclusive. Anyone with a diagnosis, in theory, can be a part of an observational study. We want to answer questions like, how do people with disease X behave over time? That's sometimes known as a natural history study. We want to know, why do people with disease X have certain exposures, different traits for the same disease? You can call those as registry studies, and if we can find biomarkers related to those traits, that would be very helpful. The second type of clinical studies, interventional studies. The research is done there to determine how an intervention can affect the person's health over time. And the intervention can be a drug, a procedure, or sometimes lifestyle behavioral changes like dietary modifications or exercise. They tend to be more restrictive because we want to select people that in the beginning of the study to be as similar as possible in their traits and disease characteristics. So over time, we can determine if there is any meaningful measurements that's going to be clinically significant at the end of the study compared to their baseline, or if we compare that to another group of patients. Now, expanded access program is a pathway for people that do not meet those strict criteria for clinical trials. And you will hear more about this later on from my colleagues on the panelists here. The last one, quality improvement, is research project that's usually done in one single center, and it involves investigation of a process or processes in a system. The goal is to improve the delivery of healthcare or to improve the health outcome for patients. So you probably have heard about clinical trials in phase 1, 2, or 3. The phases are related to how close a study drug is towards its marketing for commercial use. And that depends on the outcome of the study results. First, before any investigational study drug can be brought to be tested in humans, there has to be preclinical studies that are usually done in cells or other animal models, and they have to have a good mechanism of action targeting a biological process that we believe is involved in a disease. And when that safety data is available, then it is moved forward to phase 1. And phase 1 studies usually involve people maybe in a group of healthy subjects for the most part, and it's a small study, maybe 50 people or fewer. The main purpose is to determine if this study drug is going to be safe. And if that pans out, it goes on to phase 2. And phase 2 trials recruit usually on the range of hundreds or a few hundred patients with the same disease. The purpose is still to determine if the drug is safe and to test if it has the potential therapeutic benefit. If the results are showing favorable outcomes, then the pharmaceutical sponsor will move the study into phase 3. And you can think of phase 3 trials as a scaled up version of phase 2. And you can see at the bottom of the graph that throughout the whole clinical trials, regardless of the phase of the study, safety is being monitored throughout. And for efficacy, usually it requires a phase 2 or phase 3 studies to have the power to determine if that is the case. Then also the abbreviation there stands for randomized double-blind controlled trials. Patients or participants are randomly assigned to the active study drug arm or an inactive placebo group. And reason for that is that we want to make sure the interpretation of the results are not biased or mistakenly interpreted. And so both the participants and the investigators and the team are usually blinded to the assigned the group that the participant is assigned to. So for anyone who is interested in being a clinical study, these are the questions to ask and think about. I will not repeat it here, you can read them. Participation is always voluntary, and safety is the top priority. Dropouts from the study, that it does happen, we all know that. It unfortunately becomes an incomplete set of data for a research to analyze. So then that adds time to recruit more people to make sure we have the power to analyze the research results. So participation commitment is important. And how would you go about finding the information on clinical trials? So I'm showing you here clinicaltrials.gov. It is a website with public information that anyone can access. And you can search by a disease, by the location, or any other data fields that you choose to narrow down your selection. For example, when I just put in ALS and Florida as the location, there are 126 studies listed on this website. And if you are hoping to be in a trial, then you can filter the results by selecting only the trials that are currently recruiting. And when I did that, there are 22 trials listed on the website right now in Florida, recruiting. And the next step you can do is you can click on each of those studies. You can find out who and where you can contact to get more detailed information about the trial. On the last two slides, I will just talk briefly about the US ALS Center. We are a certified treatment center of excellence. The center was started about 15 years ago by Dr. Gooch and Dr. Vu. We have a very robust ALS clinical trial history. The research team staff is big. It was built up by Dr. Vu. We have a dedicated clinical research center. We currently are partnering with Temple General Hospital to expand our research network. And we have 3 active trials. We do have participants that are still in the study, but unfortunately these 3 trials are all close to enrollment, and they are all phase 2 trials. We do have other studies coming down the pipeline, and we hope to recruit as soon as they are open. So as everybody here knows, it takes a village to care for people with ALS, and it does take the same for clinical research. I thank patients and families for entrusting your health with our team and with all the teams, clinics across the state, and thank your time and contribution to research. And lastly, I thank the US team. Thank you.
Thank you for that great introduction. Dr. Oscarson.
Thank you, Amy. Thank you, the organizers, and most of all, thank you all for coming here and listening. So I'm Bjorn Oscarson. I'm from Mayo Clinic, and let me see. I have some slides, but you've heard about the Mayo Clinic probably. I wonder if there's anything going on there. There we go. We've been around a while. We study many aspects of ALS. I was supposed to talk a bit about what we do at Mayo, but particularly kind of lean into our collection protocols. And we all have a little piece about ourselves and kind of maybe focusing on something that we do particularly at our institution here today. We work in the neurology department, together with the Mayo Clinic neuroscience department. They are based in Florida. They are a huge department. They do basic research in ALS, trying to understand what ALS is. And if we can understand what the ALS is, we can really do something about it. We have the achieved reasonable, quite solid success for this SOD1 type ALS, where we have a clear target to shoot at. We want to have clear targets like that for all people with ALS so that we can really make a difference. So we have our clinic, we do clinical research, our clinical trials, we do this basic science, and then we try to kind of bring discoveries back into the clinic. We have never been busier. We have a great number of projects ongoing. Here are some of them that I'm particularly interested in. Most of the projects we're doing don't directly relate to patients. These are projects that are done in the background in the labs, often using samples given to us by patients, by patient volunteers. We have a new study where we're showing that we can catch ALS with a brain MRI. It's a very super strong brain MRI. We'll be presenting a lot on that this fall. We have next on the line there, received a large grant to do an expanded access program for a drug called Ibudilast. We're also doing these collection protocols as part of the ACT for ALS. The drug, the CHCG10 line there somewhere in the middle. There we have a patient with a particular genetic mutation, which we think is the cause of their disease. We've been able to get a drug made for that. That was actually something we started together with Orlando Health, and a geneticist there. And we're dosing him with this. It looks promising. It's too early to really say yet, but I am hopeful that that's going to really work for him. That's an individualized treatment. And then we're doing, we have an open phase 3 trial for Ibudilast. So again, we are looking for volunteers for that. If you are interested and you're early in your disease, you may be a candidate for that. And we do these biomarker and collection protocols. So again, trying to understand ALS at a molecular level. To do that, we're looking at, we've had success. I mean, we have this neurofilament marker that I think many of you probably have had tested on yourselves. It is a marker that we can use to make an ALS diagnosis. And it can tell us a bit about how fast someone's ALS is going. And we can use it in clinical research trials to see if a drug works or not. Now, there's still a lot of unknowns with this measure, and it's not perfect, but it's a big step forward. This is not just something that we have developed. It's really lots of institutions across the country, and I think we should give a special shout out to University of Miami for really bringing it squarely into the ALS space. We collect blood and samples for anybody with ALS who's willing. We also collect from unaffected people who don't have a family history of ALS. So we are, again, collecting a lot of fluids. That's blood. We also collect after death on those who are willing to give brains and spinal cord. And that's something which is open to anyone in the state of Florida. We do need some clinical information, other things to kind of make it happen. But generally, we are able to accept donations within the state. So, again, you can help by providing information about yourself. Again, information, samples. And if we can see the target, then we may be able to really hit it. We will keep trying based off our current best understanding. We do understand some things about ALS, and I think every drug we try, we're probably a little closer to the target. So while our track record isn't fantastic, I think that we are making real headway, and we have our first successes. So I don't know if we're some people were taking pictures. Again, I don't know if these are easy to read, but these are, again, links to some of our blood collection protocols, the protocols we do where we collect spinal fluid, and also protocols where we collect tissue. I already talked about the neurofilament, and again, I'm very curious about how this marker really will work for us. We're using it a lot in recent clinical trials. Some of the trials we started many years ago that are still ongoing are not so heavily reliant on this marker, but now we're really leaning into it. For that expanded access program that I mentioned, we'll be using this as kind of a guiding star where each patient gets their own number as they go through the project, and they can talk to their doctor about if the trial or the EAP is right for them as they get into it. And I guess that's a picture of the MRIs, what ALS looks like on an MRI with the very strong magnet camera. I'm ending early. So yes, absolutely excited about what we're working on and what we're working on in collaboration. We, on the clinic directors today, we formed the Florida ALS Clinical Research Consortium this morning. So we're gonna work more together and see that we get some real work done for you all. Thank you.
Thank you, Dr. Oscarson. Dr. Pulley.
So I'm going to take a little bit different turn here and talk a little bit more about motor neuron disease. The Euro people, they group all of the different forms of motor neuron disease. They just call it all motor neuron disease. ALS, of course, is the most frequent, most common form of motor neuron disease, but there are multiple different forms of motor neuron disease. And another one that we see, this is probably the second most common, is called primary lateral sclerosis. So there are three locations where you have motor neurons in your nervous system. One is in the brain, and those are called the upper motor neurons. And the signal starts there when somebody wants to move a muscle. The signal then connects to a second nerve cell called a lower motor neuron. And if it's for speech or swallowing or saliva control, etc., that is in the brainstem. If it's for moving the arms or the legs or breathing, those are in the spinal cord. So if you have involvement of all of them, you have ALS. If you have involvement that is almost exclusively the upper motor neurons, you have PLS, or primary lateral sclerosis. Why does that matter? It's because PLS is a much more slowly progressing form of the disease. So patients have a much longer life expectancy if they have a pure upper motor neuron form of the disease. But because it progresses really slowly, it's not easy to tell with a study, is the patient changing? And if we do some intervention, how long do we have to monitor these people to be able to demonstrate that the treatment is having an effect? Well, that's what we are doing now, is called a natural history study, as Dr. Chen mentioned earlier. So this is basically collecting data on a bunch of people who have primary lateral sclerosis. And in order to do that, it's taking a lot of different centers, because this is only about 10% of the population that we're seeing who has PLS. And so in order to collect a decent number of patients to come to any kind of conclusion, we have to have a whole bunch of centers that are doing this. And you have to follow these people and apply these different outcome measures to see, okay, which one of these is going to tell us that the patient is changing over time? Which one of these gives us the best signal that we can use in a clinical trial? There has not been any clinical trials to date in PLS. We have really no studies looking at different treatments. And one of the reasons is because people didn't really think that there was a way that we were going to be able to tell the treatment works because the patients are changing so slowly. So this is a really detailed slide here, but it just talks about some of the things. In order to be sure that somebody has PLS, they don't want it to be excessively long, so they're saying symptom onset within 15 years. They wanted people to be able to walk to some degree at the baseline. We're going to be monitoring them. They certainly could lose ambulation somewhere along the line, but in order to be included at the beginning. And they have to have a test, an EMG test, to say that we did not find evidence of lower motor neuron damage. So when the lower motor neurons in the brainstem and the spinal cord are damaged, we can detect that electrically with the EMG study. So, and they can't have other neurologic diseases that could sort of interfere in our ability to analyze what's going on. And they didn't really want to include people with significant cognitive impairment that might interfere with our ability to collect the data, things of that nature. So this is one of the outcome measures that we're going to study. I don't know what happened to that there, but so this is what's called the PLS functional rating scale. And basically, a couple of extra scoring items were added to each of, to most of the items that are on the ALS functional rating scale. That's typically a 0 to 4 scale, and now this has been transformed into a 0 to 6 scale. They're using that as the primary right now, but they're going to look at all these others that are listed over there on the right-hand side of the slide and determine, are those outcome measures even better than the PLS functional rating scale, or they're going to be more responsive, they're going to be able to show a difference in a shorter time period. Because that's the goal here, is we want to have a marker that is able to demonstrate change, and the shorter time that we can be able to demonstrate that, the better. Because, studies, we don't want to make a 5-year-long study. It's going to be hard to run that kind of a trial. It's going to be very expensive to run that kind of a trial. So if we can get a marker that is responsive and can tell us something in a much shorter period of time, that's going to be ideal. So basically, that's where we are with this at the moment. We are collecting the data. There's going to be a meeting in Montreal, Canada in December, where we're going to be getting together for a day just to talk about these patients who have PLS. It's a sub-meeting of this, what's called the Motor Neuron Disease Association meeting. There are other forms of ALS, of motor neuron disease that we haven't really touched on as much today, but so there's another thing that's called PMA, or progressive muscular atrophy, and that one is specifically affecting the lower motor neurons. So the difference with that one is that most of the time it progresses very similar to ALS. There are occasional cases of that also progress very, very slowly. And again, that form of motor neuron disease, to my knowledge, I'm not aware of any studies that have been done with it. Some of the the criteria for ALS have changed over the last few years, and things that used to be called PMA are now diagnosed as ALS. So the patients who we would have classified as PMA previously, based on their progression and based on these new criteria, they are ALS. And that these patients we've known for a long time. Most of them behave like ALS. But there are rare patients who progress very, very slowly like PLS does. So in any event, so this is the project that I'm working on currently, this PLS functional rating scale as the primary outcome in this natural history study. And I will let the next speaker come on up.
Thank you, Dr. Pulley. Dr. Weimer.
Thanks. Yeah, I'm Jim Weimer at the University of Florida, and in particular at the Fixell Institute. And like many of my colleagues, and I just want to reiterate one point that you've heard over and over again, we are all very interested in working towards a cure. And the way we do this in the clinic is, as was shown with that puzzle model, is this interrelationship, this bench to bedside to bench, just back and forth, direct relationship is a big part of what we believe in research. And patients like yourself interested in research and helping us out only helps to expand this more, learn more, and do more. You will hear, University of Florida is one of the top NIH-funded universities in the country. And we have a lot of people, just like all of my colleagues, that are doing just the basic science to try and figure that out. And some of the projects I'm going to mention are sort of UF specific because they are based on that basic science and how we can work, again, that bench to bedside. I will also be talking a little bit about the Healy, so those of you who haven't heard of the Healy and don't understand how it works, we'll be presenting a little bit of that. So we can go on to the next slide. Oh, I do it. There we go. A little bit of disclosures of some of the funding that we've used for this. And then we can start with the Healy trial. As everybody has heard, there is the Healy platform trial, goes by different names. It is based out of Massachusetts General. And what it is based on is the concept of a perpetual clinical trial. In traditional clinical trials, as you can see structured here, we basically, as the Healy team will describe it, we build this giant clinical trial infrastructure. We sort of build the stadium, build the infrastructure, we run the trial, and then we take down the infrastructure and we move on to the next trial where we build up. So this is the concept of why are we wasting all this time in building and taking down, building and taking down? Can we create these platform trials where we have a perpetual infrastructure that's constantly treating, testing. And that way, from an institution, we have perpetual coordinators, so we're not hiring and rehiring and going through for coordinators based on the studies. We've got a team that's just flowing through. And it allows for rapid throughput that will happen. And so the Healy was based primarily on that concept. And the other thing that the Healy brought as a novel concept was this idea of shared placebo. Okay, when you do a clinical trial, are you on placebo or are you on control? And everybody says, I don't want to be involved in that study because I got a 50% chance of getting placebo. I want to be involved in the study where I'm going to get the drug. So what Healy has done is this concept of shared placebo that you can see here. You can have 3 drugs that are tested in parallel and 1 placebo. So you have a 25% chance of getting the placebo. Okay, so this allows you to drop that risk from, say, a 50-50 to a 1 in 4 chance, so you're more likely to be involved in the drug. It may be 1 of 3 drugs. This also allows, as 1 drug completes the enrollment, you can bring another drug in to replace that drug. And you can see how this system can perpetually go through, and we can keep testing and testing. And this has been proven in several of the oncology spaces, and it has brought some novel interventions. It is intended as a screening process, so this is not a trial for FDA approval. It's a trial to say, do these drugs work? Do they work? Can the companies now take them on to do larger trials and get FDA approval? So most all of us are part of the Healy, and getting involved in the Healy is a very nice study that helps the science out, as well as helping identify new treatments. Biomarkers are a big part of this. The combined international nature of this trial allows us to collect biomarkers to learn more about the disease. And just this placebo arm alone, there is so much that's being learned about the natural history and designing that the Healy stands as a very good trial. Now I want to transition over to 2 different studies that are UF-based, just going through these. UF believes in a very comprehensive approach to disease at all different levels, as you'll see in this. This was something that was started in the basic science labs at UF and has now worked its way up to its first clinical trial that was going on. What we found in one particular variant of ALS, that is C9-related disease, In C9, there is an abnormal protein that accumulates. It's called RAN proteins. I won't go into the specifics of that, but that protein accumulates. And then in the animal models, we found evidence that one drug in particular, metformin, can reduce the production of those. So this is for C9 specific. Frequently I get questions, does metformin work for ALS? The answer is it is not being looked at in ALS. It is looking at C9 ORF72 mutation ALS, a genetic form of ALS with this abnormal protein that we're looking specifically at. Does it impact it? If you're trying it in generalized ALS, in general ALS, it needs more testing to identify it. And this is what we're doing with this, the goal being this particular protein, PKR, is reduced. We have animal data that's listed here showing that you can significantly reduce with metformin. You can even improve behavior in the animal model of it. And we went on to a human trial. We have completed that human trial, and hopefully before the end of the year, if not the beginning of 2025, we'll have all the results in and be able to say, is metformin worthwhile going on to another study? Lastly, what I want to touch base on is this is something that, again, all of us feel very strongly at, but we've worked on. Certainly in the past, we've presented data that we did with Dr. Tabor Gray and Dr. Lucatelli's group, where we looked at speech and swallowing. And this is kind of what I call the low-hanging fruit. These is research studies on things we can do that can improve you here and now. Exercise components. What can we do to try and help you to develop things that can be rapidly implemented and put into the clinic? It's something we all believe in, and we had done this looking at breathing exercises, which has now pretty much become the national standard to start doing these breathing exercises because of work that we have done in the state of Florida as a collaboration to develop the benefit from those and pushed it out. Very simple, very safe, making a huge impact. In another group, what we raised the question of is breathing. You go into the clinic and what do? We test your FVC, okay? And we say, this is your FVC. Well, FVC is complicated. It's like a global rating scale of the lung. What's going on? Can we improve it? Can we impact it? And what we found is this little trick called acute intermittent hypoxia, which sounds terrible, but it's basically high altitude training. Okay, what we do is we put a high altitude training mask on you and we vary your oxygen level from Gainesville to Denver to Gainesville to Denver. And as you can see from this, we do these by intermittently doing it. You can see the slope going up, that it can gradually improve it. And then in this study in question 2, it improves, we believe it improves some of the neuroplasticity in patients with ALS. So it can impact some of the neuronal control to the lungs. And we're not looking at a home run, a huge improvement, but a minimal, safe intervention that has the potential to help sleeping. And now we're looking at medications that have the potential to also enhance that in parallel with this. So working in combination with certain medications as well as the AIH treatment, can we potentially lead to improvement in the breathing of ALS patients, in the plasticity of the lungs, and allow for improved lung function? That again, it gets back to that sort of something that's easy, simple to do, that can make an intervention right now. This is very much an academic study. It is ongoing right now. It is not a clinical trial. Everybody gets treatment because we're trying to understand the mechanics of how does it improve, what does improve, what biomarkers can we look at before we begin a much larger study. And I'll finish a little bit early too.
Thank you, Dr. Weimer. Dr. Locatelli.
So good afternoon, and I know that I'm between your questions and the panel. So what I'm going to try to do is flip it a little bit towards your side and give you a sort of continuation of your journey about living with ALS, and when and how you can choose where and how to participate in one of these trials. I just want to point out here, you see a picture of what we call the CCR, or Clinical Collaborative Research Building. So it's a collaboration of research. And this is collaboration with other groups in the building, collaboration with other centers in Florida, collaborations with HEALY, which is very natural. 80 sites in the country are collaborating to run that study. And then collaborations around the world to trying to include everybody, as the goal of finding the cure is not one center, but everybody together. Collaboration also means that we need to involve pharmaceutical companies so they can bring their compounds and help us to market them later on. And it also includes you as the patients and the caregivers to be part of this study. So it's just the patients, is the researchers, is pharma, is the academic institution. So this is really a big team effort that we need to get all together to figure this out and find the cure. And here's this slide that I borrowed from the University of Miami. Michael Benatar has really looked at this carefully because he has done a lot of research in genetic ALS. And one of the things that happen if you have a genetic form of ALS, sometimes your family members are going to be tested, and they're becoming positive, but they don't have any symptom. So you see here at the top of the panel A that the disease can begin at a certain time, but before that, there's already something about ALS that is not yet presented to you. And down there, we can sort of subdivide that disease process in what is called the pre-symptomatic phase. And then sometimes there will be a prodromal. Something is not right, but you just kind of clearly pointed it out. And even though a pre-manifestation phase in which you can have markers like neurofilaments or other markers that say, well, something is beginning to occur. So you have a gene positive, maybe a marker goes up, but you still don't feel anything. Maybe something is not right, but it's beginning to occur. So at that point, you begin to see that the disease is already in somebody's body and is going to manifest later on. And then we go into, okay, there's a definite symptom, and we have a clear problem. We have a diagnostic delay for about a year plus, almost everywhere. And that is really a big problem, because if we're going to intercede in that treatment, the sooner that we put it, the best results that you're going to get. And then finally, you get to a center, EMG, you do a lot of studies, and everything is negative, and then somebody says you have ALS. And then you start progressing, and you go through different phases until you get 4 or 5. So this is something that is progressive. And here's when the timing, particularly when either you have a family member with a gene that you're kind of waiting, or you being diagnosed, that's when you realize that for you, time is very important. So here's where research is changing in trying to find that cure. In the past, we tried fast, now we're trying faster. So the design of the studies today are modified to trying to get outcomes sooner. It used to take about 2 years, 2 year plus for the design. So it took 4 to 5 years to end a study. So it took a long time to actually say, well, this didn't work. Now let's do it again, and let's do it again. Today, the study's been shortened to 6 months. Maybe there's some changes to be 9 months, maybe 6 was too short. So we are shortening it to trying to find go, no-go answers. In Europe, they are using neurofilaments to actually test these compounds and say, okay, it's likely to work or not, and then move forward. So the idea is to do research that is finding results a lot faster now than ever before, because time, as you probably know, is an essence. My time goes much slower than your time, and we understand that. And so we try to design studies. And one of the things we've done really well is we put the patients at the design table. And they're telling us, what is their recommendation about design. It's just not you participating on the study as a patient, but participating as a sponsor of the study and trying to make recommendations on how to run it, how to do it, and so on and so forth. But here, with this model, I just put here in your journey, how you can sort of participate in these things. So we said, well, there's a pre-symptomatic phase. So if you're a family member that has a gene you're positive, you might enroll in a natural history study and trying to understand when these neurofilaments are beginning to say the disease might be turning on. And there's actually one of the studies that is, as soon as these filaments may change, you might be treated with this medication specific for that gene, the SOD1. So even almost before you start getting any weakness, the doctors will be able to treat you right on the first spot. So at the very beginning. Data presented by Tim Miller, which is one of the pioneers in the studies with tofersen, tell us that if you're treating with this particular medication that involves about 1 to 2% of people with ALS, if you treat at the very beginning, there might be even some reversal of symptoms. So if you know you're gene positive, to be there with the drug when you kind of at the beginning, that's essential. And we have over 10%, maybe 15%, depends, what population you read, can be genetic. So understanding those genes and treating very quickly is very important, because that's when you're going to try to make sure that you stay very stable or even potentially reverse it at the beginning. But natural history studies go from the different phenotypes, and like PLS and Dr. Pooley was talking about. So pretty much from the preclinical phase to later on, you're going to be studying the disease progress. Now, and the green bar is where these phase 1, 2, or 3 studies that we heard from Dr. Chen at the beginning, that you can participate. At that point, in those phases, you need to have a diagnosis. So it cannot be preclinical, I think, at this point. But particularly the double-blind phase studies is when you're going to have a placebo arm on it. And then these drugs, we don't know completely the side effect profile. So you cannot be very advanced. So your breathing needs to be fairly good. So your mobility needs to be fairly good. And most of the time, you need to be able to swallow the medication. Depends on the administration. But usually, you cannot be very advanced. And actually, the FDA would not let people take placebo if you're advancing your journey. So that probably would not be an option if you're very advanced. So you have to pick a different study. And second is, you have in the blue is the expanded access program. I'm going to mention a little bit more of that. So if you don't qualify for the green bar, and so the double-blind study, you may qualify for an early access program. And then at the later stages, I put BCI, which is brain-computer interfaces. So there's complete different kind of research, but if you really get in that stage, there might be techniques in which computers can interface with your brain and you can still communicate and doing things. So there's a lot of research going in that phase as well. And in this slide is a kind of a summary of what expanded access program is. So it's the possibility that you can early access to an investigational drug that is not yet approved by the FDA. So if you don't qualify for the double blind and you want to participate, what can you do? Well, just a few years ago, there was nothing to be offered. Maybe a natural history, maybe something to understand the disease, collect information for the future. But there was no option you can access to these medications. So if you're not eligible for an ongoing clinical trial, you probably qualify for an EAP program. This is a research, so you have to sign consent. You have to be willing. You have to volunteer for this. You have to sort of request it. You need to know that the safety and the efficacy of the drug is not fully understood because it's been tried just in a few patients, not many. So you're taking a risk, but you take the benefit that you're going to be not in a placebo, you're going to be receiving the medication. So you're at kind of more of an advanced stage. You're going to be on the drug. You're not going to be taking the placebo. And also it's important because now in the past, these programs were to 1 or 2 patients. Now the government is sponsoring these programs well, so they are going to be at no cost to you. So they're free. So you don't have to have a copay. You don't have to go pay your visits, so on and so forth. These programs are at no cost to you. So this is the summary overall of the expanded access programs. We right now have 2 ongoing expanded access programs in the Institute. They're very popular and they're well received, and people are demanding and trying to come up with this. As you know, Dr. Orkerson is about to get a very nice grant, and he's working really hard to open another expanded access program. So these programs are becoming quite popular. And so there's another option that you can take in addition to the FDA medications. You can take these drugs as we wait for the double-blind study of the same drug to finish so we know whether they work or not in terms of the efficacy. These studies are not looking for efficacy. They are helping to understand the safety of the medication. And with that, I want to thank the team, which I know you make the effort to come here today. At the Nova Southeastern University Kathy J. Hassman Center, we opened just about 2.5 years ago. We have now a very robust team. We are doing things differently since it's a brand new from the ground up center. We're about to open a 7,000 square foot, fully dedicated for care or research, 100% on ALS. We are focusing on patient care, but particularly our main goal is finding the cure. So we're going to establish collaboration and creating a center that hopefully will make good changes and good contributions so we can find the cure that we're all looking for. So without any more speakers, I think we're going to sit down and then take your questions. Thank you very much.
Thank you, Dr. Locatelli. Wow, what an impressive group and what a great series of presentations. As I was listening to your remarks, I was so impressed. I kept thinking, I'm hearing collaboration. I'm hearing urgency. We're got to speed things up. We got to find ways to cut through what's delaying process. And I'm hearing innovation. The Healy platform trials, expanded access programs, these are all great news to hear. Prior to working with the home office, I just have to tell you, I worked in care services in Southwest Florida, still live here, and it's very near and dear to my heart. And in care services, I went to clinic, and I know how important the clinic relationship is to the people that are living with ALS that we serve. I also facilitated support groups, and there is a very dear person. His name was Trimble McCullough. He was a drummer who developed ALS, and he had to stop drumming because of respiratory issues, not because of weakness in his hands. But he used to, at every support group meeting, he would say, "You got to have hope. You got to have hope." And I just want to thank the panelists for bringing us some hope today. This is very encouraging. So thank you all.